Thank you, Everine, for your general introduction. And thank you, Paolo, for showing us how to go the way forward. Now, my
Thank you.
task is actually to look back and use some very classical scientific approaches to critically ask whether some of our maybe favorite water models or features are actually realistic. And I think that's also one job of the World Water Lab, as Everine has explained, that our common denominator is we're all scientists and we're really curious to understand. We don't want to sell anything, so we try to be as unbiased and critical as possible. And I know that is really hard sometimes. And yet, some of us have shared the experience of spending a lot of time following some phenomena, doing research into a dead end. And part of the World Water Lab is to rule out theories that have outlived or been outdated by now. So I would like to take you on a journey that started 2019 when I was on a sabbatical and became really interested in water, visited the World Water Conf the World no, just the Water Conference organized by Jerry Pollack. And that's where I got to meet most of you who are now in the in the audience and part of the book writing team. So I'm now going to flood you with a lot of data. Don't worry about it. I don't want to shower you too badly, but this is just to substantiate my claims because you have to be very careful when you criticize colleagues' work. Um, I try to be as clear as possible about our own evidence. And this is also there to illustrate to the book writing team that I've done part of my homework and got all the data together. So, the topic I'm going to assess critically is that of solid water that can exist at room temperature. It is called Xerosydryle by the team of Vittorio Elias and Roberto Germano and their co-workers, and has been published in many articles now over the last decade and more. And in fact, it is all based on a very nice concept that you must all be familiar with, which is that by Jerry Pollack on the exclusion zone. He called that the fourth phase of water. And what you see here, that a piece of polymer or any other material, when it is immersed into a solution or suspension of small plastic microbeads, forms a thin layer on its surface, which grows here as a function of time and becomes visibly thick, up to 0.5 millimeter. And this is a genuine phenomenon which Jerry has tried to explain in terms of a model of honeycomb layered water molecules, which I doubt, but in the end, it's only a water model of a permanent structure, which is something Paolo told us not to believe in. But the concept is that this water in the so-called exclusion zone has certain properties, and they are, for example, characterized by a fingerprint of UV vis absorbance of 270, 280 nanometers. And it would be curious to find out more about this. That is the classical view, asking, is it possible to extract it or somehow scrape it off the surface? And in fact, this exclusion zone is very general. It has been observed, and we have reproduced that in our lab here on the surface of numerous materials. Here you see a muscle fiber forming an exclusion zone. Here you form a polyacrylic acid gel, forms a nice exclusion zone. Even an optical glass fiber forms an exclusion zone. And so the idea was by our colleagues Vittorio Elia and Roberto Germano, whom I'm just going to refer to as the lab in Naples, in the city of Naples, is that this fourth phase of water, this exclusion zone water, could be harvested by like literally scratching it or gently leveraging it off the surface and accumulating it. This accumulation process can take several months, and it can be monitored, they published, by increasing conductivity, usually by decreasing pH, and they claim that the water that they harvested, the fluid, is similar to what Pollack described as easy water, has a higher density and higher viscosity and this fingerprint absorbance. Now, the most curious point was when the Naples team then lyophilized this harvested water, and they call the dry residue Xerosydryle. In fact, this Xerosydryle is supposed to consist of just water molecules, H2O, but they are solid at room temperature. And that is really something very spectacular. You see here on these round bottom flasks, 2 examples that they have published in good journals, that they actually can harvest a residue from surfaces of various nature. And so when I came along to see these results, I was fascinated and wanted to do some solid state NMR analysis. That is because my team in Karlsruhe is a biophysics group. We work with biomolecules and we have special access to nuclear magnetic resonance that can be applied to solutions, but also to solid materials. So we wanted to look at the proton spectra, look at the structure, something solid, not recommended by Paolo, but find out what holds these water molecules together. And that was the start when we tried first to prepare our own IPW. Oh, I didn't explain that. That means iteratively perturbed water. And that's the fluid system. And when you lyophilize it, then it's called Xerosydryle. So to prepare this material, you take some sheet of a technical membrane, for example, Nafion has been famous for that, but also some natural fibers, immerse them in a petri dish of water or in a large container, and wait. On the right here, you see that some supposedly easy water, or what people might call wrongly coherence domains, form right on the surface as solid entities. Then they can be scraped off by gentle agitation, and then you remove the solid material from the container, dry it overnight, and immerse it again in the next morning. And you do that for many weeks until this solution is literally full of easy water. Then you can lyophilize it. And you can do that with the starting materials I mentioned: Nafion, silk fibers, sheep wool, paper filters, cellulose, cotton, cannabis. Many different things have been published. Even just pumping water through peristaltic pumps is supposed to give the Xerosydryle. The analysis follows conductivity changes, pH, light scattering, thermal gravimetric analysis, mass spec, scanning electron microscopy, atomic force microscopy, circular dichroism. We wanted to contribute the NMR. So this really seemed to be a very established topic. However, we did not manage to make any Xerosydryle under sterile conditions. We followed the method of Elia and worked very carefully in closed container, even in a clean bench. But, well, we did find a decrease in pH over weeks. But that's not surprising because the material we used as a starting material is Nafion. It's a fluoropolymer that carries very acidic groups. So naturally, when you immerse them in water, they will give off protons and acidify the medium. But we were expecting to see an increase in conductivity, but we did not really find anything. So after lyophilization of these IPW samples, nothing remained. We had no material left and tried that many times and did not manage to do this, working very carefully and sterile. Until one day, our PhD student Lakshmi had a cold, and lo and behold, we saw a nice gradual increase in conductivity and thought, well, now we're on the right track. But just to be sure, we looked at the aqueous sample here on day 33 and under the microscope, and it was full of bacteria because she had a cold. These were streptococci. We had a microbiologist in the group, so this was very disappointing. And finally, we had been working online with Elia and Roberto to get their advice on how to prepare these samples. And finally, we had to ask them that they should, whoops, this was one too far. That they should give us one of their samples. But I remember now, I included one slide which could be interesting for people doing homeopathy or working with very high dilutions, because we wanted to check how sterile can we actually work over a period of days and weeks using a sterile bench, a clean bench, or an open lab bench. So on the left hand, you see here Nafion treated water, 12 days on the lab bench in closed containers that were only opened when you had to insert or take out the Nafion. And it's full of bacteria after 12 days. That's not too surprising. But we then went to these sterile conditions in a clean bench where we had done our experiments. And even there, working in closed container, pure milk water is full of bacteria, and by eye you can hardly see a difference in the two types of milk water. So this is a word of warning to ourselves and to everyone. Even clean benches are not sterile. And please keep in mind that the Naples group had worked in a lab bench in open containers that were not even covered for weeks on end. So with this warning, which we kept everybody informed, we finally asked them to send us some of their material because we did not manage to do it. And here you see what we received from them. Whoops, the mouse isn't working. Several samples prepared from silk, sheep wool, etc., and also from Nafion, technical membranes, although the latter sample was only a very small amount because they say that is the most difficult material from which to prepare Xerosydryle. And here you see a sample, a photograph of some brownish material. It was actually a bit crunchy and hard. Here you see a special NMR tube. It is submitted to high-speed rotation, so it has a little vortex here. And I actually had 4 people working on the NMR analysis, the microbiological analysis of these IPW samples and the Xerosydryle samples from Naples. I will show you some solid-state NMR data looking at all the nuclei that we might be able to find in this water, supposed H2O, but also that could emerge from biological contaminants. We did the protein analysis, microbiology, microscopy, and in the end also some conventional liquid state NMR that will prove my point. But first, this is one of the most exciting slides that we started off with. I told you we used solid state NMR, special magic angle spinning techniques, looking at the Xerosydryle derived from silk, sample from Naples. And we were expecting to see proton signals in a water-like environment near a shift of 4 to 5 ppm. That's where you would usually see proton signals in water, liquid water, ice, etc. But there was not much. There was hardly anything. A bit broad here. But remarkably, we see here signals of aliphatic and methyl groups. Interestingly, these signals are both in the Xerosydryle sample, but also in the starting material. So in fact, these two spectra look very similar. So we were wondering maybe some of the starting material made it into our Xerosydryle sample. And so to be sure, we did a protein analysis on SDS polyacrylamide gel of the many different Xerosydryle samples we had received. And this is a molecular weight ladder, which allows you to look whether there's any protein present in the sample, which gets stained. And in fact, all the abundant samples had multiple protein contaminations, many bands. So yes, there is definitely a lot of protein in there and not just H2O. Interestingly, only the Nafion sample was not available because it is so hard to produce and they did not have much material for us. So let's look at the NMR again. Now we chose the carbon nucleus because carbon is so characteristic of organic material and biological material. And you see here a simple NMR spectrum of the different Xerosydryle samples from cotton, cannabis, cellophane, silk, sheep wool. And they have characteristic signals of proteins. These are the carbonyl signals, that's characteristic of protein, and the C alpha atoms here, but also of CO carbohydrate signals, as well as lipidic signals, aliphatic ones, over in this region. And in fact, most samples look pretty similar, but in fact that they contain both proteins and sugars, carbohydrates. But interestingly, the silk sample, the Xerosydryle from starting material of silk, was a little bit more simple. It contained seemingly only protein here, but no carbohydrate. So we focused initially on the further NMR analysis of the silk sample from Naples. And here you see a detailed comparison of the Xerosydryle from silk and from the starting material, the raw silk fiber, which the Naples lab had kindly also sent to us. And in fact, these two spectra look quite similar, but they are not truly similar. You see, these peaks are shifted compared to that. So it is not the raw material that constitutes the Xerosydryle. It is similar. Now, let's think about the structure of silk fibers, how they are composed in a simple biochemical view. You see here a picture of a silk fiber that has a core of fibrin protein. This is what gives it the strong, dural, tense, what do you call that? The tense character. And it's consisting of the protein called fibrin. This protein has an unusual amino acid sequence. It's rich of alanines, glycines, and gives unusual NMR spectra. The two core fibrils are, however, coated by another protein called sericin. And this is a gum-like protein which makes the fibers a bit sticky so that the silkworm or spiders can make their nets. And now we can look at the separate NMR analysis of sericin on the outside and fibrin on the inside, because you can buy the material or prepare it yourself by washing the fiber. And here is a comparison of the raw silk fiber, the complete bulk, and the pure fibrin, this protein that gives it the tensile strength. And now you see these two spectra are very similar. In fact, the starting material of the raw silk fiber is essentially dominated by mass by the pure fibrin component inside. And the sericin cover is only a very thin layer. You don't see much of it in the NMR spectrum. However, you can buy sericin, and we looked at commercial sericin, but also at spectra that are published in the literature, and compare this now to the Xerosydryle NMR spectrum. And now we see there is a complete correspondence of the Xerosydryle and the sericin signature, which means that the Xerosydryle is essentially the sericin that has been scraped off in the procedure of preparing the iteratively perturbed water and then the lyophilite to harvest this material. That was a bit disappointing. We told Ilya, etc., but maybe there was still some other material on top of that, although we did not see protons that could make up H2O. Now, as we had all the different Xerosydryle samples, we continued with another sheep wool sample and the cannabis and the paper filter, etc. But just to give you an example of the sheep wool, again, the Xerosydryle sample gives essentially the same spectra as the sheep wool, the purified sheep wool, except for some additional sugars or carbohydrates that are presumably further contaminants in the Xerosydryle. And it is known that sheep wool is consisting of keratin, a protein to 33%, but also a lot of so-called sheep sweat. It's called suint. It's a microbiological metabolism in the fleece. The activity of the microbiota leaves their mark on the unwashed wool. And so we thought, how can we detect microorganisms with NMR? And we decided to look at the phosphorus spectrum because phosphorus in DNA and RNA and ATP, our energy molecules, and just the phosphates floating around in the cell, that is very characteristic of life. And what you see here is a phosphorus NMR spectrum, solid state NMR, of the dried sheep wool sample and of starting material, pure sheep wool. And you see, indeed, the Xerosydryle has accumulated lots of phosphate, lots of phosphorus, even though sheep wool keratin has hardly any phosphorus. So the Xerosydryle sample must be contaminated with stuff that is contaminated from bacteria, the suent, and maybe extra bacteria that flew in during the long time of sample preparation. Now, we continued with yet another sample from Naples, and that was the Xerosydryle prepared from naphthalene. This is the fluoropolymer with the acidic groups shown here. And since fluorine is a very sensitive NMR nucleus, we thought, wow, let's look at the fluorine NMR spectrum of the solid Xerosydryle. And interestingly, at this point, we hardly saw anything. A very weak fluorine NMR signal compared to a huge signal in the starting material. Now, this told us the Xerosydryle sample that was prepared from naphthalene was not full of organic material, and it was not full of starting material. It was full of something else, but that something else was not H2O. So what could that be? At this point, we started a microbiological analysis with lots of microscopy, and this is published, so I will go only through this very quickly, because we did light microscopy on the liquid sample, the iteratively perturbed water, IPW, initially, and we saw microalgae that sometimes grow in strings. We also saw much smaller bacteria, streptococci, as in the sample where somebody had a cold, and we saw lots of inanimate non-biological debris. Microscopy of this kind had not been done by the Naples group yet. And interestingly, when we subjected the same sample, the fluid sample, under the fluorescence microscope, we saw these interesting dimeric features and tetrahedral structures. A nice tetrahedron is seen here or here. And it is known that diatoms have autofluorescent properties. You can just shine light on them and they fluoresce. And these diatoms are unicellular algae which have strong silica wall. So could it be diatoms? In fact, it is known that diatoms are everywhere. Ernst Haeckel, a very important traditional biologist, actually drew, when the light microscope was invented, beautiful pictures of these diatoms. And you see here tetrahedral or planar structures, these dimeric type of structures. These are due to the silica shells that look a little bit like petri dishes that can be intercalated into one another and surround the algae, the mononuclear cell inside. And in fact, the city of Naples is so close to the sea, so it is not surprising that algae living in the sea just get washed out in the air and by a breeze land in the lab of the research group, drying their sample preparation sheet of naphthalene or wool or silk or whatever was used. And in fact, a study that we found from 1964 was that in Texas, dust was collected, and in 1 liter of air, 180,000 diatoms were found, even in the middle of the desert. So diatoms are everywhere. You cannot avoid them when you work in open labs, and even under the clean bench, they find their way into your sample if you're not extremely careful. So next, we did some scanning electron microscopy. You see just some examples here. Microalgae of several 2 to 200 micrometers are seen here nicely. Bacteria are much smaller, but you can see them here also as little crumbly stuff surrounding the larger bacteria, larger algae, and a lot of strange objects, probably inanimate dust and debris, was also detected. And here you see some electron microscopy images of the Xerosydryle. Please note, the previous pictures were just from the fluid after perturbation, and this is now the Xerosydryle, where things have agglomerated and they're much harder to discriminate because they've been freeze-dried, cells will pop and material will stick. So you see here something which is probably of biological origin but clumped together, and this here looks like a synthetic fiber of a few hundred nanometers, micrometers diameter. Right. To be sure that our analysis of organic matter and silica shells was correct, we did some attenuated total reflection infrared analysis of the fluid sample from Naples, prepared by Nafion, which had no starting material contamination. And we see here all the stretching and bending vibrations of silicate tetrahedra, of amide bonds, characteristic of proteins, and of aliphatic groups, possibly also some fats and amino acids. But interesting, we could confirm there was no Nafion fingerprint vibration, which is of the sulfonyl groups. So let me get another point across, which is why we also are not convinced that the Xerosydryle has any substantial solid water in it. The Naples group published some thermogravimetric analyses from Xerosydryle made by Nafion, and more recently also Xerosydryle obtained simply by pumping water through a peristaltic pump again and again. And this stuff is really stable up to high temperatures. And interestingly, when we looked into the literature, a protein spectrum of bovine serum albumin, standard protein, has a very similar structure to the contaminated sample from Xerosydryle made by Nafion. You see these two. And in the case of peristaltic pumping, we are pretty sure, just looking at the literature spectrum of silicone, that from the peristaltic tubes, silicone has rubbed off. This is a known phenomenon. It's actually a known problem in pharmaceutical industry. And so the silicone itself gives a different thermogravimetric analysis to the protein, but that was also found for the Xerosydryles from different origins. So I think that can explain really everything. Now, therefore, we can conclude that the Xerosydryle samples are actually a mixture of lyophilized biological and also non-biological contaminants. The three types of contaminants that are important are the starting material that gets rubbed off from silk and wool, etc., paper filter, the biological contamination, bacteria, algae, diatoms, was found especially in the Nafion sample, but also in the others. And there's also a lot of non-biological contamination, like dust particles or polymer fibers. So I'm not going to read this slide for you, but I guess the meeting is being recorded, so this is just for reference. People can look where the Naples group may have gotten carried away a bit in terms of their interpretation and by not doing enough controls. And even though I'm really convinced that they did the best they can and they collaborated with a lot of other groups, it was presumably very hard to interpret all the different kind of data that came into them because they were not experts on so many different methods that were used to characterize their Xerosydryle. And they did not have the means to do the microbiological analyses that we did in Karlsruhe. So up to that point, it was all pretty clear, but then came another publication from Jerry Pollack, who's well known to all of us. And he said, yes, he himself was able to produce with Abhay Sharma and others solid water at room temperature by harvesting the EZ, the exclusion zone water. And that again was a point that suggested that perhaps the Naples group was showing something real that had just been overlooked due to a huge amount of contamination in the samples that they worked with. And if I get another few minutes, Everine, please, I would like to go through the careful analysis of Pollack's data because
Yeah,
their,
quick.
thank you, their paper was actually very carefully crafted. They used very reproducible description of their method. They used very short incubation times of only 10 minutes as opposed to several weeks. So they carried out control experiments. They were very self-critical and even analyzed their solid water with mass spectrometry and said, well, maybe there's a few lipids in there. But let's see what we can find out by looking carefully at their data. So we can exclude, by all means, biological contamination. Oh, I lost my mouse. Sorry, my mouse is gone, but you can see on the slide. Yeah, here we are. The biological contamination is improbable due to the short incubation times, and the non-biological contamination of dust is also unlikely because they had only 10 minutes before they collected their samples. But what about the starting material? This starting material in the Pollack paper was ghee, which is clarified butter. They also used Whatman filter paper, and as a third starting material, they used Nafion sheets. So let's see carefully if we can find anything in ghee that, let's go back, that has the characteristic fingerprint signature of 270 nanometers. Yeah, this is what Jerry Pollack used to show that there is easy water, because easy water is supposed to have also the fingerprint here at 270, 280 nanometers. So the first thing we did was to buy some ghee and dissolve it in a solvent that has a suitable UV vis range to allow us to detect this component. And indeed, in ghee, there is a strong component or a significant component at 270, 280 nanometers. This could be due to protein, but there is known to be just a very small amount of protein left in ghee because it's clarified butter without the protein. But it could also be due to unsaturated fatty acids, because these polyunsaturated fatty acids have an absorbance at 270 to 280 nanometers. So clearly, ghee butter contains lots of different fatty acids. So we thought this might be a clue as what may have happened in Jerry's samples. And here we reproduced the production of his solid, easy water according to their protocol. From the ghee, you first take a spoonful into a glass container and melt it. Now, from the molten solution, you transfer 1 milliliter into a new, fresh, clean glass container and let it solidify 20 minutes in the fridge. And then you add 1 milliliter of water, let it incubate for 10 minutes so that just the surface touches the water for 10 minutes. You siphon off the water and do the analysis. So this is very clearly explained. And then you check for the absorbance. You can do control samples. You can do mass spectrometry. And that's what we did. So in fact, we could reproduce the liquid water that was siphoned off the ghee surface. It had a massive 280 nanometer peak. Is it protein or is it polyunsaturated lipid? Interestingly, when we lyophilized the samples, this peak became much smaller and was lost. This is also something that was written in the paper. But now, without going into the NMR analysis, we did all the reference spectra, also in deuterated solvents to look for the proton signals, etc. And we prepared our lyophilized GEZ sample. And you see here a small interesting set of signals seen here, which when we enlarge it, shows that this extracted water from the surface of ghee contains lots of unsaturated methylene and methine groups, which are characteristic of lipid acyl chains, especially of polyunsaturated lipid chains. They seem to have been enriched in the easy layer on the Yi sample, on the surface. And turning to the literature, it is in fact known that, for example, docosahexaenoic acid, which is shown here with lots of double bond, has a pronounced absorbance at 270, 280 nanometers. And this polyunsaturated fatty acid is highly abundant in milk. It's very healthy for pregnant women to drink milk. And because of this DHA fatty acid, this was written in the paper of 2019. So this interpretation is fully consistent with our NMR data. And now let me try to explain or describe how it comes that we enriched so much unsaturated lipids on the surface of the ghee sample. And this has something to do with this melting and solidifying process that was used by the Pollack group to prepare the easy layer. I think they did it to make samples really reproducible. But in fact, what must have happened is that a lipid mixture in the molten sample starts to cool down, and when it nucleates, the saturated lipids will nucleate out first because they have the highest melting point. So when you wait and the sample solidifies further, there's so-called crystal growth of these saturated lipids. They aggregate with each other, and in the end, on a little droplet or on a surface towards water, they get pushed out, or they push out the unsaturated oleic acid and polyunsaturated fatty acids because they have a lower melting point and they don't like to sit side by side in a quasi-crystal lattice. And in fact, as water scientists, you're very familiar with this process as to what happens when a droplet of water freezes, which is contaminated or just containing some solutes. Ice does not want to accommodate any solutes in a crystal structure. So when water droplet cools down, ice nucleates, a small ice crystal grows and excludes the solute from the ice, pushes it onto the surface of the water droplet. Yeah, so the surface is now enriched of the solute. In the case of the lipid mixture in the melting state, this would be enrichment in the unsaturated oleic acids and polyunsaturated ones. So with this analogy, let me very quickly go just through the analysis of the Whatman paper filter that Pollack also used to produce this so-called solid easy water. But I hope to show you that this can also be interpreted by very simple other contaminants. So we reproduced actually the 280 nanometer fingerprint by squeezing Whatman paper filter, led to a turbid suspension, and was hard to measure due to scattering because this paper filter is so full of filling stuff. But we saw that absorbers. However, it is known that any kind of paper really likes to soak up human sweat, and human sweat is there on every healthy being that touches or handles surfaces. This is a test I know from medicine that allows to detect the evaluation of a perspiration disease. A sick patient who doesn't perspire well shows only a very weak fingerprint, or in this case, a footprint on filter paper, which has been stained with ninhydrine. It's a spray that gives a purple reaction to amino acids. But a healthy person just putting their clean foot on a piece of paper filter leaves proteins from their sweat on that paper filter. And so we believe that this absorbance at 280 would probably just be due to the sweat proteins on the Whatman paper filter. And finally, since this later paper from Pollack also used Nafion as a starting material, we very critically looked at the data whether this 280 nanometer signature was actually there with Nafion. And in fact, this is from his paper. The ghee paper shows the absorbance here. The Whatman filter paper shows the absorbance here. But the Nafion easy shows hardly any signal here. So there's not really much to it. And after lyophilization, you see here, there is actually no fingerprint signal. So I think they did not see any fingerprint for EZ, only a little bit of wishful thinking, maybe attributed a little bump to that, but I don't think there is one. So let's be critical. And I can summarize, but I don't think the time allows, that there can be many different types of contamination of the starting material, of the environment, and of bacteria. And depending on the samples that we measured, we got different types of contamination. In the Nafion sample from Elia, we found mainly bacteria and dust, whereas in the Xerosydryle samples from silk, sheep, cellulose, etc., we found mainly the starting material. In the Pollack sample, which we reproduced from ghee, we found polyunsaturated lipids absorbing at 280 nanometers. When we looked at his filter paper, we found proteins that absorb at 280 nanometers by ninhydrine staining. And the EZ lyophilized sample does not actually show the EZ sample. So I think we've cleared up all these questions, showing that it really is a dead end to keep up the idea of Xerosydryle and of solid easy water at room temperature. Now, again, I'm not going to read this out to you, but for the documentation, it will be published pretty soon, and then also in our book. We did, Evelyn, should I go through this or go back to finish now?
I think we finish. Yeah, sorry.
Yes. So thank you for your attention and thanks for your patience. We're all taking a bit longer.
Thank you, Anne. So this was hopefully just a view on how we can help each other to interpret what we find when we are collaborating from different disciplines and with experts who know different ways to test. Because Anne spent a lot of time doing all these things to understand what somebody else did to be able to have a good argument for another case, but most of us won't have that. Anybody wants to ask Anna a question? Yeah.
Alex here. Thanks, Anna, for really going in depth into these analysis. I'm sure everyone told you we had some of these samples in Heidelberg, and we were able to pass them through the analytical chemistry labs that told us there was a lot of oxygen, nitrogen, carbon there that was very suspect. So, but we weren't able to go in any way, in any depth that you did. So that's very, very interesting. Just before we came here, we were in a meeting in Paris for the FMRC group, and Michel van Wassenhoffen has told us some of his experiments where he's dehydrating samples. And he's not going into like any claims of Xerosydryle like here, but he does see residue. Most of it is, according to him, due basically to the carbon dioxide forming carbonates and then being as a residue. Is that something that you've seen yourself? I'm just wondering from your perspective, what's the role of CO2? You're using ultra pure water, so in principle it's degassed when you get it, and then it will absorb CO2. Will this form, in your perspective, especially if you succuss and it interacts with the walls, you have many phenomena where it could form residues without having any of the particular processes that you mentioned, biological or otherwise. What's your opinion?
Yeah, thank you for that question. It's actually very intriguing. And I think we will need to go back to our carbon-13 NMR spectra to look for any CO2 or carbonate fingerprints. I'm afraid that the strongly contaminated samples containing so much starting materials are completely dominated by the protein NMR spectra, so it may be hard to detect traces of CO2. But the Nafion Xerosydryle may give us a chance, made from Nafion, because that was not contaminated from starting material. But then again, the biological contamination was significant, it did not give as much material as the rest, according to the Naples group. But therefore, we did not have a lot of sample to look into minute traces of CO2.
I know you haven't had those samples, so I was just asking for your opinion rather than any specifics. Also, these were homeopathic samples, so highly above Avogadro, ultra pure dilutions in sterile conditions. So we don't expect any of the phenomena that you just talked about. I was just curious about your opinion about these carbonates, and I know you haven't looked at it in detail. So.
But if you can get hold of some of these samples, we'd be very happy to do some carbon solid state NMR on them.
Sounds great.
Yeah. Because also another thing is, for me, this group of water lab, okay, is to try to provide some error before the publication of the paper, because you know. You make a really good job and really fantastic to show that some results we obtain are not the reality, okay? If it's possible to check that before the publication, it will be really more comfortable for us, because it's very easy for the contradictors to say you make a really bad work, okay? So it's your work, and all around the world we have to pay attention about this key problem.
Thank you. In fact,
Just
we
a short
had hoped...
No, go on.
Yeah, we had hoped to have Vittorio and Roberto on our paper as co-authors, because we kept in touch with them and showed them all our results and had their advice how to continue. But they declined. And I think they're still not convinced of our data, which puzzles me a bit.
About homeopathic remedies, just a comment. As I told you yesterday,
A little bit louder. Yeah.
oh, sorry.
Because this is not this.
That's correct. About the homeopathic remedies or potentized remedies, I think that actually the truth stay in between, in the sense that we have both impurities and dirtiness due to the process, but at the same time, the configuration of the water, of the liquid, are creating some non-trivial pattern that actually acts as a healing or stuff or stimulus for the body. The error would say that to associate the two things. So the configuration of water are due to the potentization, and beyond them maybe you can also have dirt, dust or whatever. So at least this is something, this perspective could be kept in consideration, and thank you.
Okay, so Leonie, a guy wants to give you something, but you want to ask also a question. He needs to leave at 12, so we have 5 minutes.
Just a second. No, just to remark, it made me think about the Maria Olga Kokornadzic work, in which copper salts were used as a mask to define patterns of crystals, and so after many repetitions and systematic controls, etc., she could publish 2 papers in scientific reports showing that indeed the patterns of these masks changed according to the homeopathic potency inserted there. So maybe what we are seeing here is also a kind of mortuary mask using bacteria or other solutes, etc., but not the water itself. Maybe the interpretation is wrong, but we are seeing one thing and thinking in another thing. Maybe it could be like this, just a thinking, okay? Just to think about. So can we stop? Because
just one minute only.
Do something. Here, you can.
So this is my book. I am presenting to our dear, my mentor, Leonie. This is my book on Practical Guide on Veterinary Homeopathy. So this is a practical handbook from A to Z. We have covered every disease in this book. So this is on for veterinarians of India. Thank you so much. Please. Please.
Oh, yeah,
Yes.
Okay, so Anna, sorry, this needed to be done before 12, but maybe there's more questions. Yeah, there's more questions.
Sweets from India for all of you. They are like there. Please accept them. And I'm leaving. Thank you. I have a very nice company with you. Thank you so much.
Okay, thank you. Bye-bye. Okay, you want to, or you? Okay. Okay.
Oh, I need
You have a question?
Yeah, that's fine.
He's going to ask the next question.
After.
Okay. All around with
Your taxi is waiting?
Okay. Go ahead.
It's really our...
Oh, sorry. Yeah, it's only for Anna, but he's... Okay, thank you.
Thank you.
Okay, see you. Good luck.
Thank you.
Okay.
It's real pleasure to gather here to deepen our knowledge. It just came one question in my mind. Classical remedies, in classical medicine, we have a lot of side effects. And it just came to my mind, what do you think if we will put the drug in the water, and we are talking about memory of water, that water will take the information, I don't know how we talk about, and that the patient will drink this water without just in the mind that we will diminish side effects of the drugs. I'm just thinking if we will just put classical drug in the water, leave it for, let's say, 5 minutes, 10 minutes, 1 hour, and then the patient will drink this water without residue. And if it's possible that the memory of water will take the information of the drug, and if it will have any effects of the drug.
Sorry. Would this be dissolved then or not? So if
dissolved and.
So you would just drink the solution of this.
Yes.
I think that in this sense we can only expect a hormetic effect. So let's say hormetic effect is, I think, when this is diluted to 10 to minus 2 or so. So it may come, sometimes not, perhaps. But I think that for memory of water, it is important this succussion process, and theoretically. Yeah. Okay. Just.
Yeah, I mean.
Isotherapy, so the same thing. Exactly. In isotherapy, you have many homeopaths use it for detoxing antibiotics and different treatments that have been abused in heavy metals. Then high dilutions, homeopathic dilutions of these compounds are used to detox these compounds. So it's a relatively well used. I mean, here I can pass it on to the doctors.
But we have to process not only to dilute, but we have to make a dilution.
The homeopathy.
Condensation.
Okay.
And we have some effect, not just like that, because we have a 30 CH, 15 CH, not
Yeah,
before.
I understand. One thing,
Yeah.
one thing are homeopathic remedies, but on the other hand, I would just like to know, because we are talking about memory of water, if you put medical drug, classical drug in the water, if it will have any effect.
So, I mean, people didn't hear you just now, but
I understand. I understand. We have two things. One, homeopathic remedies and diminishing of side effects. Let's say medrol. We are taking cortisone on 9-CH. This is one thing. But on the other hand, I would just like to know, because we are talking today of memory of water.
I can answer something, and we are also in our group where we have people who are doing infopathy and things like that. So we we can talk about different things. They seem to work, and it's valuable to try to understand that. And I think it's a good question. We are talking about all kinds of different mechanisms, but indeed there are people visiting the water conference doing these types of things. So there's a lot of things. Does anybody have a question for Anna still, or are we? Yeah, Michael. Okay, and that's the last.
Yeah, thank you, Anna, for a very extensive work. It's really nice to see good quality work. Yeah, I have first a comment. I mean, I also wrote it in the chat. I spent an entire year looking for this absorbance of 280 nanometers. I never found it. Maybe it's because of my own incompetence, but we never found it. And I think that was because, and I spoke later to one of the guys in Pollack's lab, who actually did this work, and he used this flow cell because he used a normal UV vis, and then he used the flow cell. I mean, you need the spot of the light, you need to kind of put it in 0.11 millimeter, and there you start to hit the surface. The surface was apparently he used some UV curing for this, so that was kind of my head explained. So that was just a little comment of my own work. I wasted an entire year. Another thing is that if you have this Xerosydryle and EZ water, it should look like ice. So why not? Shouldn't you expect an ice spectroscopic signature from this? I mean, why do they claim all this? I mean, for me, it's clearly it's biological pollution, no? So what is your comment on this?
Well, that was the starting question. We thought, well, nobody has done the solid state NMR of the ice fingerprint in NMR. Let's just do it because we have the spectrometer. It would be like a one-day experiment, and lo and behold, we prove that it's real. And it took many years to prove that it's not. Because the ice signature on the proton NMR spectrum just wasn't there. And we even tried oxygen 17 NMR, which is a very tricky experiment. And we didn't see anything either, except for just oxygen and proteins and esters.
Okay, we can continue the discussion later. We will thank everybody this morning.